So we are going to move on to Dr. Theresa Kotanchek, CEO of Evolved Analytics and also co-chair of the National Materials and Manufacturing Board of the National Academy of Engineering. So Theresa, please take over.
Okay, great. Thank you, Krish. Today, I'm going to address the modeling of the manufacturing process in regenerative medicine, but by way of background, I first became involved with this due to interaction with Krish about 4 years ago when he came to the Georgia Tech Manufacturing Institute External Advisory Board with the enthusiasm of what was ahead in cell manufacturing. And with the recent work that had been done in road mapping, laying out the critical challenges and the potential opportunity to bring a community together to address these challenges. As has been articulated through a number of the presentations, the need and current lack of reproducibility standards, quality by design, what are the critical quality attributes, what to measure, what indeed is good in the midst of the heterogeneity, how to measure the critical quality attributes, how to predict safety and potency, and address simultaneously and in manufacturing with lower costs. And as we all know, getting a workforce in place to actually exploit that. The great news was following that, the CMAT was formed, the National Science Foundation Engineering Research Center and Cell and Manufacturing Technology, with the vision to transform manufacturing of cell-based therapeutics into large-scale, lower-cost, reproducible, and high-quality engineered process for broad use. And the beauty of the ERC is that it is indeed bringing all of the critical stakeholders together, both from industry and practitioners, from the regulatory side, patient advocate, to address the integrated requirements that are indeed necessary. And to Amy's point of yesterday, to address this language franca that we need to bridge between those of us that may not have come from the foundation of biologics, but are more on the informatics and analytics side to our manufacturing industrial engineers. So CMAT is positioned to address these key challenges, particularly as applied to quality, cost, speed, and agility. The examples today that I'm going to highlight really speak to the opportunity and initial work concerning multivariate CQAs, multivariate CPPs, and what that can do in leading to real-time monitoring. One of the key programs that is part of CMAT is on dynamic sampling platform with the objective of indeed being able to achieve the objective of reproducible predictive measurement and assay technologies. We need to develop non-destructive inline closed system analysis using real-time sampling, sensors and imaging tools, and being able to utilize that in a robust way, both not on the discovery side, but in the process development. For those who are unfamiliar with CMAT, I'm not going to go through the details of the three-tier diagram. Chris is, of course, the expert in this, but simply to call out the power of the program is a recognition that there are key deliverables needed in new scientific knowledge, in the delivery of new tools and technologies to build the integrated systems, and the recognition that these building blocks can be applied to different test beds. And the example that I'm going to show today is specific to T-cell test bed. So in this case, we'll build this out, is each of the projects are critically addressing and linking into the cross-cutting engineering system, recognizing that it is an integrated system, and what do we need to address in order to support the legs of that system and build it in a way that we can use it in a both understanding and for predictive purposes. So the specific project I'm going to talk about is the one around the variability. And this specific example is in reference to degradable microscaffold microcarrier cultures, which the work that has been done is showing the potential for expanding for more central memory in lymph node homing T cells. So the question is, how can we control that, and how can we use that, if you will, as a test bed for development of the workflow to enable multi-omics characterization, and do so in an unbiased modeling of the end product for early predictive signatures of quality during manufacturing. The project has been structured both to understand the variants as well as establish CQAs and CPPs that are predictive of potency, safety, and consistency. In this case, the specific example were really a two-phase prong. The first was examining the experimental process parameters and doing optimization of the input process parameters for the microscaffold, looking at responses of viability, CD4, CD8, naive memory attributes. In this case, we utilized the data modeler software, which is using evolutionary computing to do the predictive optimization. And with that, we were also able to identify regions for further improvement on extrapolation. So we executed a sequential adaptive design of experiment to further test, validate, and move into an optimized region that had not been part of the original study. Of course, in this case, to Anne's earlier comment, everyone is talking about big data, but in a lot of our early program work, we actually have smaller data sets. And the beauty of being able to use the evolutionary tool set is that we can manage through that and deepen our understanding. And the phase 2 multi-omics integrative approach, also looking at the impact of the microcarriers, but now process parameters, but also included secretome profiles as well as the metabolites. In this case, we merged process, secretome, and NMR data, looked at total lives CD4, total lives CD8, the ratios of CD4 and CD8 with the objective of looking at, can we in fact control both simultaneously? And in this particular study, we also did a comparison of different machine learning analysis along with linear modeling to look at the ability for us to move forward. Let me move. Oh, my slides are not moving forward.
Can you?
There we go. Okay. So before we get into that, in terms of, I want to pause around the modeling options and the recognition that when we're looking at different algorithmic approaches, sometimes people lose track of the fact of their underlying assumptions, and those underlying assumptions really are tied to what we believe we know and what we don't know. In particular, what do we believe we know about driving variables? If we, as well as the model structure itself, if we know the driving variables and we know the model form is linear regression may be perfectly fine. If we know the driving variables and nonlinear regression parameter estimation. If we know the driving variables and we don't know model form, we have a variety of powerful machine learning tools. But when we are in a situation where we aren't entirely sure of driving variables nor model form, we can use the power of symbolic regression, both to identify using the evolutionary process, but also give us explicit, transparent models that we can test. And the evolution handles the symbolic regression model development automatically, generating novelty from the data. So it gets back to yesterday's discussion. What data do you have, and exploit it? Solutions are found by simulating natural evolution and selection, and rather than biological species, in this case, being created in mathematical expressions. Novelty is generated by the competition of the fitness, in this case, of accuracy versus complexity. As I said, the beauty is that you do generate explicit, transparent, interpretable models, and as such, it can be used as an augmented intelligence tool for hypothesis generation, where the domain expert can indeed examine those solutions which emerges most accurate. So not all models are created equal. The focus is in on that collection of models which are giving us the highest accuracy, lowest complexity, and we can exploit that. We can also exploit the collection of models rather than worrying about the model in terms of what are the collection of models telling us? What are the potential driving variables? How many variables are actually required to get accurate models? We don't want to drive to overfit when we're utilizing. So we purposely are looking at how we can narrow, if you will, that variable set often. What are the variable combinations, their distributions, and what are emergent meta variables that can give us further insight? This discussion of big data versus big insight is an important one that was highlighted in Anne's presentation. And obviously, if we have 9 million records and all but 10 are equivalent, we're not going to gain as much information and insight in the early development process as we are from looking at the diversity of data. So this point is very critical in that we want to look at all data, all that in that context of what you believe is good or bad data, bad being not giving you the result that you anticipated, so we can exploit the understanding of that. And furthermore, we can take the power of the collection of models to build what are called ensembles. We utilize the diversity of that collection of accurate fit models to indeed generate our trust metrics. So just as a very simple representation of this, if you had the simple trajectory of a ball and you only enter the first half of the data set using the genetic programming, within seconds you would identify the prediction and you could focus in on that collection of models that are extremely high accuracy, extract the diverse set, and generate an ensemble where the ensemble's mean trim is giving you a projection, but you're also being warned, go carefully. You have not exploited the space. This is the area where a collection of further information could be most valuable. If you were to take the same type of data and look at other machine learning comparisons, they may or may not give you the accuracy of indicating where you can and cannot trust if you were to move forward. Also, the power of the ensembles is in the active design of experiments or active learning, where we can take the ensembles that we've built, and in this case, the same trajectory, but if you only had the beginning and end points of a trajectory, were to build the ensembles, it would do very accurate where there's data points. It would give you a projection, but also, again, warn, beware. It drives your understanding if experiments are cheap or expensive, and limited time, where you are most beneficial to go collect data, to gain and glean additional information, reanalyze and lock down your understanding within that space. This is the whole concept of trustable models are the foundation of active design of experiment and how one can utilize it to target the next critical experiment. So the bottom line is, in terms of CMAT, we've applied these types of approaches. We have indeed been able to use the design of experiments and active design to optimize conditions for maximizing memory T cells, and also the design of for CD4, CD8, the ratios, the design of the parameters for the microcarriers and what their interactive effects are. We've also been able to merge the multi-omics data set and do the comparison across the various learning machine learning methods in terms of accuracy of prediction, including all of the omics in the analysis, looking at earlier time periods. In this case, day 4, it was a 14-day collection of data over a period of time, but looking at process parameters plus NMR process parameters at time period 4, 6, and also with the cytokine. So the machine learning was performed and was effective in both identifying cytokine dependency as well as looking at their interactive effect and the ability to predict total live memory, CD4 and CD8. In addition, we were able to use for early prediction just using the NMR, both on ethanol and lactate with the microcarrier characteristics to look at optimization. And we can do that simultaneously, as well as if the objective were to optimize one versus the other. So what are the key factors that are driving? How should we design the system? We're utilizing this workflow approach to be able to extract that information. So now we can engage in identifying what are the appropriate targets and design for the dynamic platform. As I noted, for active design of experiments, it is extremely powerful, and system integration is absolutely paramount for the active learning process. There is a natural workflow of integration of data sources, statistics visualization, data consolidation, looking at model space descriptors, doing the modeling, deploying it to the subject matter experts, doing the validation analysis, identifying the next set of experiments, and feeding that loop for continued learning. Moving forward, my call out is these approaches are not limited to the types of problems we've looked at. The question is, what is your question? What is your analysis objective? Whether it's in the discovery side, if it's in your development platforms, if it's in the validation, the same methodologies can indeed be applied. And the questions that you're trying to ask. In some cases, you're simply trying to understand which variables matter, what combinations are most useful, what are important meta variables. Is there a need to do accurate prediction? What can we control in real time? For optimization and development, can we build robust emulators for what if and design? And can we simultaneously optimize multiple objectives? And can we build this in an active learning mode? Also, equally important, outlier detection and assessment, and the extrapolation of what is the extrapolation power of the models being generated. So with that, I will close with the call out of what data do you have? My encouragement is exploit it. Exploit both the experimental and manufacturing knowledge that you have when something isn't generating the target response, because that is powerful, similar to the patient discussion of who's responding to and who is not, and really understanding the differences between those two patient groups. So with that, I will close.